people logging in, so we will start in maximum 2 minutes, just to give the time to everyone to fix any possible technical problems. Again, good morning. So now we are ready to start. My name is Francesco Nicoli. I am from the University of Ferrara in Italy, and together with Dr. Mkunde Chachage from Tanzania, I will co-chair this session. So this is the fourth appointment of the One Health Knowledge Café, which is a project born in the framework of the Center for International Health of the Ludwig Maximilian University in Munich, and with the support and thanks to the support of the DID, again from Germany. It's a project that brings together 11 scientists from different countries, so from different geographic location, and also from different disciplines, because the idea of this project is to try to understand better the COVID situation, even putting together different perspectives. And mainly, this project is mainly focused on the dissemination and communication. The first 3 webinars were aimed to describing the situation in some specific countries, and with this webinar and the following ones, we will rather focus on some hot topics or topics we think are of relevance for the COVID-19 situation, like the one of today, which, as you know, is the COVID vaccine. So before introducing the speaker, a few housekeeping rules. So I am asking everyone attending this meeting to keep muted and the video off. Of course, this does not apply to the speaker. Of course, the session is being recorded and will be then available on the website of the Center for International Health. We will have a question and answer session at the end of the presentation. Anyway, we encourage people to write any possible question in the chat box. The schedule is very tight, as you see. I'm also asking the speaker, please, to stay within the 10-12 minute frame for the presentation. And so I think we can really begin the scientific and most interesting part. So I'm very honored to co-chair this webinar, first of all, for the topic and for its relevance, but also for the list of top presenters that accepted our invitation. So we are very grateful to them for being here and for sharing with us some of their time and their experience and their knowledge. The first speaker I'm going to introduce is my co-chair, so Dr. Mkunde Chachage from the University of Dar es Salaam. She's located in Beja in Tanzania. She is working since years in the immunology of infectious diseases, and especially in the context of HIV and of co-infections. Today, she will introduce us she will set the stage for the other presentation, introducing some basic concepts of vaccinology in the context of COVID. So please, Mkunde, we are very happy to listen to your presentation.
Thanks, Francesco. Can you allow me to share my screen, please?
Yeah, I think you can now.
Can everybody see it now?
Not yet, Mkunde.
Mm-hmm. Let me just see what the problem is. So share screen. How about
Yes.
now?
Yeah.
All right, great. Well, thanks, Francesco. Hello, everyone. So, as Francesco said, my main task today is to level the ground and set the stage for the upcoming speakers by quickly giving an overview of what vaccines are and what it takes to make them. So, basically, all vaccines work the same way. They work by training our immune system to combat a pathogen when it encounters one. And when you receive a vaccine against a certain pathogen, it's like you're receiving a mock infection for that pathogen. And an instruction will be sent to your immune system to generate or to evoke an immune response that is somewhat similar or even sometimes stronger than that would have been invoked by the real pathogen that you're trying to prevent. So it's some sort of a mock infection that is introduced to you without making you seriously sick or ill. And what a vaccine also does is to train your immune system to generate memory response so that when you're ever encountered by the pathogen that you're trying to prevent, the response against it will be quicker and effective in a way that it will be stopped, the pathogen will be stopped and destroyed even before it gets a chance to establish an infection. So there are different types of vaccines that are out there that have been made, and they essentially serve to introduce the whole pathogen or part of it into our bodies in order to trigger the right amount of immune response. So you've got some vaccines that are in form of a whole pathogen in a killed state or a weakened state. For example, polio vaccine or MMR vaccines have been made in this way. And some vaccines use only fragments like proteins or fragments of the pathogen, while some newer ones that are coming now in use or they're in human trials use pieces of genetic material that come from the pathogen that you're trying to prevent and introduce it into our bodies. And that in order to train ourselves to temporarily make the proteins of that pathogen so as to stimulate the required immune response. And these genetic materials can be DNA or RNA from the pathogen, whether it's a virus or whatever pathogen that you're trying to prevent. And they can be delivered directly or packaged in what we call a vector, which is usually a bacterium or virus that is in a weakened state and not harmful to us. So I hope you can see my cursor. Under normal circumstances, it takes something like 10 to 15 years to develop a vaccine. It's a very costly process with multiple stages from the discovery phase in the lab to optimization of it in animals, and then to clinical trials in humans, which have a couple of phases. So there's the phase 1 that checks for the safety of the product in small groups of people, usually healthy, followed by phase 2, which usually checks as to whether the product generates or mounts an immune response. And then bigger trials, that's phase 3, that looks at how effective the vaccine and how safe it is in a wide array of people. And then the vaccine moves on after all these human phases, it moves on to the regulatory phase where it seeks approval from the agencies for use and marketing, which of course, it can also be a lengthy process in itself. So as costly as this process is, not all vaccine candidates make it to market. So you've got like 1 in 10 candidates that would make it to licensure, and a lot of other candidates will be phased out or filtered in the earlier stages. So in this pandemic, we've seen vaccine developers working in a speed that we've never seen before. So what they're trying to do is to achieve all this in a shorter time period, and they put something like 12 to 18 months. So how are they doing this? Well, what they're reporting is first, instead of starting to do things from scratch, they're actually using similar platforms or technology that have been used into making new vaccine candidates that have been used, for example, for making vaccines or vaccine candidates against pathogens like HIV or Ebola. And these technologies are really good. They can be employed at a very good speed and precision. And also what they do is to actually overlap these clinical phases and run them in overlap. So you've got a preclinical trial which overlaps with the phase 1 trials. And once the doses of the phase 1 trials have been given, the investigators will check for the safety of it. And if it shows to be safe, then they simultaneously start with the phase 2 to check how immunogenic the product is. And once they've given all the doses of phase 2 and it looks to be safe and immunogenic, then they simultaneously begin with the phase 3 in some of the candidates. Now, if you're following up on this topic, you see that they have already started with the phase 3, where they now check in bigger groups for the efficacy of the product. And in the middle of it, if they see that it's efficacious and it's safe, then they move on to the regulatory phase for approval for the product to be used. And after that, of course, they would have to, start working into manufacturing it at a very high speed, very short time, to make sure that it gets to the end user, to the people who need it very quickly. So this is not a small task. It's something that's very tough. And the stakeholders involved in this have invested a lot of resources, human capital, and time to make sure that this happens as planned. And we are hoping that it indeed will happen as planned. So as much as we are all desperate for a vaccine to ease this pandemic, shortening the time period to achieve, to get a vaccine should not come at the expense of the safety of people, and it should not go without rigorous processes of review from the scientific community, from the independent regulatory authorities, and even from engagement with the communities that are going to benefit from the vaccine. Of course, there are issues that also have to be considered, like issues of transparency and equity when running these clinical trials. And now I'm talking especially on the bigger ones that some candidates are into, the phase 3 one. We are really banking on the people involved in this to really run this in an ethical way, to make sure that indeed the vaccine that they're testing, they will report whether it really is efficacious in a diverse group of people, given that we know that vaccine-induced immunity can vary with age, with sex, with ethnicity, and even on whether or not a person has some other underlying health conditions. There are issues that we have to be mindful of course, as well, issues of accessibility of the vaccine once it becomes available, or even acceptability. And this is especially true for vaccine candidates that are employing platforms that have never been licensed for human use before. And there are other unknowns, like how long will, like whether or not the vaccine that will be efficacious enough will have long-lasting effects. Unfortunately, we'll come to know this when the vaccine has been administered. I'm sure these issues will be discussed by the next speakers, and maybe even in our discussion, the Q&A session. So with that, I'd like to end, and I'm looking forward to the next presentations and later on the discussion. Thank you.
So thank you very much, Mkunde, for this very clear overview. So it's really my pleasure now to introduce Professor Florian Krammer, who graduated from the University of Natural Resources and Life Sciences in Vienna, in Austria, and then he moved to the Icahn School of Medicine at the Mount Sinai in New York, where he works since 2014 as an independent PI and is currently a professor of vaccinology. The work of Professor Krammer focuses on the understanding of the mechanism of interaction between antibodies and viral surface glycoproteins and on translating these into novel vaccines and therapeutics. So Professor Krammer will give us an overview on the current state of COVID-19 vaccine development.
So thank you for the introduction. I'm trying to share my slides here. And I think you should be able to see them now. Yeah, so I'm trying to give an overview of in general, of how far they are in the pipeline. I'm sure Barney is going to go into more detail about RNA vaccines in his talk. But I wanted to start a little bit about immunity, because before we talk about vaccines, we need to basically understand what we want to achieve with vaccines. And so if you look at SARS coronavirus 2, at the particle, which I'm showing a schematic here, we have a number of important immunogens here. We have the nuclear protein on the inside of the particle, which is pretty immunogenic. And then we have the spike protein on the surface. And the spike is really the main target of neutralizing antibodies. And because of that, it has become the main target of vaccine development, as you will see. So the spike is this surface glycoprotein that the virus has, that it is using for binding to the host cell and then getting into the host cell. And very important is this receptor binding domain, which is shown here in red, which binds to the human ACE2 receptor. And so we know that if we get infected, we get a lot of these antibodies against the spike protein, and we know that they are neutralizing. We also know by now that we get, through natural infection, a pretty good T-cell response against the virus. And that's a strong CD4 T-cell response and a relatively weak CD8 T-cell response. And we also know that even mild infections usually give you a robust antibody response against the spike protein. I'm showing data here from Mount Sinai from our plasma screening program. Here on the left side, you see the titer distribution of people who are positive for the virus. And we have these 5 different titer categories. 1 to 80 and 1 to 160 is low. And about 7% of people develop these low titers. 1 to 320 is moderately high, and about 20% of individuals develop that titer. And then 1 to 960 and 1 to 2880 is high, and about 70% get into that titer range. This is based on about 20,000 people. And you can see this is screening over time. A lot of people, even with mild infections, make a robust antibody response. And the important thing is these antibodies really correlate, the antibodies to the spike really correlate with neutralization. And that's shown here, where we have neutralizing titers on the y-axis and the ELISA titers on the x-axis. So we, as humans that are infected with the virus, really mount neutralizing antibody responses. And there was a lot of discussion initially about how long-lived these antibody responses are. There's more and more data now that shows that it's a pretty normal antibody response that peaks after infection, then declines a little bit and then stabilizes. And that's what I'm showing you here from one of our studies, where we enroll people about 1 to 1.5 months after their infection, and then we follow them longitudinally. And what we start to see is very, very stable antibody titers. And that's important because we think that the vaccines will behave in a very similar way. And of course, if the antibodies would go away after a short period of time, that would not be helpful for vaccines. And we also got a first glimpse of a correlate of protection, maybe a first indication that neutralizing antibodies could be protective. There was a recent study that was published in JCM of a fishing vessel where, to start out with, 3 individuals on the boat had neutralizing antibodies, and then they went out on a trip, and there was an outbreak. Actually, 82.5% of the sailors were infected with SARS-CoV-2, but the 3 people who had neutralizing antibodies did not get infected, which suggests that they were protected. Of course, those are small numbers. It's just one study, but it's nice to see that. And that's what we hope we'll see in larger studies and in the vaccine trials as well. So let me just jump into the vaccines. We have already heard about different types of vaccines against SARS-CoV-2, and I'm basically showing these different platforms here. Again, here in the middle, you see the actual virus and the spike protein. This is what I showed before. And there's now different ways of making vaccines. You can either inactivate this, grow this virus in cell culture and inactivate it, this is shown here, inactivated viruses. There are several that are very far in clinical development in phase 3 trials, mostly in China, but there's also a candidate in India, and now there's one coming up in Germany. You can attenuate the virus either by passaging it under unfavorable conditions, and that's kind of the old school way of doing it, or by genetically modifying it. And there are some candidates that are in clinical in development, but very far away from clinical development, unfortunately. Then we have viral vectors, and they can be replication competent or replication incompetent. And the candidates that are furthest along right now are replication incompetent. This is, for example, CanSino's Ad5 vector, but also the Chachage vaccine that is now developed by AstraZeneca. But we also have replication competent vectors. For example, measles is used right now, and that's developed by Merck. And that is an interesting candidate as well. And then we have recombinant protein vaccines, which are all shown here. This could be a recombinant version of the spike. And Novavax, for example, has a candidate that's very far already in clinical development. You could use the recombinant RBD on its own, but you can also make virus-like particles. Medicago, for example, is making those in plants. We have DNA vaccines where just a vector is used with the spike protein on the mammalian promoter, and those vaccines are in clinical trials right now too. They seem to progress a little bit slower than other candidates. And then, of course, we have the RNA vaccines where messenger RNA is encapsulated in lipid nanoparticles. And 2 of the front runners, the candidate by the Vaccine Research Center and Moderna, and also the Pfizer candidate are based on this technology. And I'm sure you will hear more about that from Barney. And this is just an overview. It's actually slightly outdated of where all these different candidates are in the clinical, in the development pipeline. We have now more than 140 candidates in the preclinical phase, according to the WHO. A lot of them are recombinant protein vaccines, but there's also a lot of viral vector vaccines, virus-like particle vaccines, RNA vaccines. So you have the whole variety. What we are lacking a little bit is live attenuated vaccines, as you can see here. And a lot of these vaccines are already in phase 1, phase 1/2 trials, phase 2 trials, and even phase 3 trials. And I'm saying this is outdated because we have now 10 candidates in phase 3. Novavax just started a phase 3 with a recombinant protein vaccine in the UK. And so this is extremely promising. We know that we need a lot of vaccines. It's very likely that 2 doses have to be given per person. Ideally, we would want to cover the global population, which means something close to 16 billion doses. And no one vaccine producer can actually make that much material. And also, it will take a very long time to make all these vaccines. So if you have many candidates that make it through phase 3 and eventually get licensed, that's of course helping a lot with satisfying the global demand for a vaccine. So diversity is really key here, and also global distribution of manufacturers is key in this development. So it's, as you see, it's less a race against each other in terms of vaccine candidates. It's more a race against the virus. And I wanted to kind of give a little bit of an overview of data that is already available. So what I'm showing you here, it's a very busy table, is data that we have from published data that we have from different candidates in non-human primates. I'm doing a comparison here, but you have to take this with a grain of salt because many of the assays are different, challenge doses are different, and even the monkey models are different. And what we can see here is we have Sinovac, which is a Chinese producer that makes an inactivated vaccine. They tested their vaccine in these monkeys. They actually gave it 3 times. They ended up with relatively low neutralizing antibody titers, and then did a challenge with 10 to the 6 TCID50 or SARS-CoV-2 intratracheally. And what they saw is that the lower respiratory tract, the lung is protected, but the upper respiratory tract wasn't really protected. So basically suggesting this works in protecting from disease, but you might still get infected. A similar vaccine was tested by the Beijing Institute of Biological Products and Sinopharm. And again, this was in this case given twice. The neutralizing titer ranges are similar and the outcome was pretty similar. So the lungs are protected in these animals, but the upper respiratory tract is not. Then the AstraZeneca vaccine, which is a chimpanzee adenovirus that expresses the spike, was also tested at 1 dose and 2 dose regimens. They get a little bit higher neutralization titers after the second vaccination, so it goes up to 1 to 160. This vaccine candidate also induces a T-cell response, and what they saw is that at least if they give the vaccine twice, you also get protection of the lung in the animals, but you don't see much protection of the upper respiratory tract. Janssen, so J&J is also developing an adenovirus vectored vaccine based on AD26. They did a 1-shot vaccination regimen in monkeys. They actually compared many different vaccine constructs, and their best construct actually protected the upper and the lower respiratory tract after 1 shot, but the challenge dose is lower here than for the ones that we just discussed. And then we have the 2 RNA vaccines. Then we have the RNA vaccine of Moderna. I'm not going through this in detail because Barney will present it, but what you can see is that you get very high titers of neutralizing antibodies in these animals, and you also get complete protection of the lung in the high dose. And then we have Novavax, which is a recombinant protein vaccine, and they get astonishingly high titers, neutralizing antibody titers in these animals, and get also complete protection in the lung. And for the high dose, they also get complete protection of the upper respiratory tract. Again, they use the lowest challenge dose, and it's a different non-human primate species. So as I said before, it's really hard to make comparisons here because the systems are different. If you look at published clinical trial results for phase 1 and phase 2 trials, we also have a number of candidates here. Sinovac published their results in humans. They get neutralizing antibody ranges with the inactivated vaccine, similar as what they see in non-human primates. In this case, after 2 vaccinations. One interesting finding was here that they get lower neutralizing titers in older individuals, which is something we need to keep an eye on. CanSino tested their adenovirus vaccine at 2 doses. They get relatively low neutralizing antibody titers, and they also seem to see a lot of side effects that are driven by the viral vector. AstraZeneca, the Chachage vaccine was also tested in humans with 1 shot or 2 shots. They get higher neutralizing antibody titers, specifically after 2 shots. Again, with this vaccine, the side effects, what I mean by side effects is things like fever, headaches, or fatigue were relatively high, and that's what you would expect from a viral vector. Again, I think Barney will discuss the Moderna data in more detail, but what the first human trials showed is that similar to the non-human primate model, you see after the second vaccination very high neutralizing antibody titers in humans. And you also see relatively good CD4 T cell responses. And then Pfizer also tested an mRNA vaccine. They actually tested two candidates in parallel, 162b1 and 162b2. B1 is just the receptor binding domain encoded by the mRNA. B2 is the full-length spike. They tested different doses, made comparisons, and they get results that are similar to Moderna. So they see high neutralizing antibody titers after the second dose. Basically, they also looked at younger versus older individuals, and they see the same that Sinovac is seeing as well, that older people have a harder time to make high neutralizing antibody responses. For the two mRNA vaccine candidates, it was interesting that the first vaccination didn't induce really a lot of side effects. The second vaccination did. Again, these are not concerning side effects. It's more or less a response to an immune response to the vaccine. And then we have Novavax, and similar to what they saw in the non-human primate model, they get very high neutralizing antibody titers in humans. And they also looked at T cell responses and saw a decent CD4 T cell response. So what I'm telling you here, and that also summarizes the non-human primate data, is that there's a lot of vaccine candidates here that actually are very promising, that induce neutralizing antibodies and to some extent also T cell responses. And we don't see really severe side effects for any of them so far. And so that's very promising for moving forward. One important point that I wanted to make from an immunological perspective is that, and that's something that's reflected also in the non-human primate data, our respiratory tract is protected by different types of antibodies. The lower respiratory tract is protected by IgG, which corresponds to the serum titer, basically, of IgG. But the upper respiratory tract is mostly covered by secretory IgA1. And these antibodies are really hard to induce by vaccines that are injected. And this is shown here. But they're usually induced if you give an intranasal or mucosal vaccination. And I just wanted to point out that all vaccines that are in advanced clinical development right now are injected vaccines, and it's not clear how well they will protect the upper respiratory tract. And that we could end up with vaccines that protect very nicely from disease, but might allow for some asymptomatic infections in the upper respiratory tract. I think that's something to keep an eye on and also something to look at in clinical trials. So just to conclude, in terms of natural infections, we get more and more data after initial scares that the immune response to infection is relatively solid and that the immune response looks normal. We have an initial strong increase in antibodies driven by plasmablasts. Plasmablasts die approximately 7 days after they were induced, and the IgG that they made declines a little bit because there's not much, it's not continuously produced. And so you see a decline of titers after a few weeks and months. And we already see that these titers stabilize at a certain level if you go 3, 4, 5 months out, and that is driven by long-lived plasma cells. So it looks like a very normal immune response. The $1 million question right now is if the level of antibodies that are secreted by these long-lived plasma cells is above or below a protective threshold, and that's a very important area of research right now. We also see that the antibodies that bind to the spike protein correlate well with neutralization, and it's unclear yet what role T cells play in protection. Some data from non-human primates and vaccination suggests that at least CD8 T cells don't correlate with protection, but I think this is not conclusively answered yet. And in terms of the vaccines, we have several candidates that induce strong neutralizing antibody responses in non-human primates and in humans. It's currently unclear if those responses protect humans and what quantity of antibody is needed, and that's why phase 3 trials are conducted, and we'll have the answer hopefully soon. Protection from lower respiratory tract infection in the monkey model seems to be very solid for a lot of these vaccines. The protection in the upper respiratory tract is often just partial. And so it's not clear if these vaccines will protect specifically long-term from infection in the upper respiratory tract. Another point that I wanted to make is that it's currently really hard to compare across vaccine candidates. All the assays are different. For the non-human primate model, the models are different, the challenge doses are different. So it's really hard to actually tell you which one is better and which one is worse. And we also don't know right now how the vaccine-induced immune responses compare to natural infection in terms of protection and longevity. And because this was mentioned earlier about the longevity of or duration of protection, I think if we get vaccines that protect for a few years, and we should be fine, even if protection wanes after 2 or 3 years. A booster dose could be given. This is done for many different vaccines, and I don't think that would be really problematic. So at this point, at least, I'm not very worried about waning immunity after vaccination. And with this, I'll stop, and just wanted to thank the team here at Mount Sinai and my collaborators across the globe for their help in the recent in the last 9 months. Thanks.
Thank you very much, Professor Krammer. We ask you to give us an overview of the current vaccine candidates, and now I think our knowledge about it is much wider than before. And on top, it was very interesting also the immunological perspective, what is known and what is not yet known on the immune correlate of protection. That was just great. So I'm asking everyone if they have any question to write in the chat box. We will then try to pose all the questions at the end of the session. For this reason, I am also asking to speaker if they can stay in the time frame of the 10-15 minutes, so to leave some space for the question at the end. And so now it's really my honor to introduce Dr. Barney Graham, who is Deputy Director and Chief of the Viral Pathogenesis Laboratory at the Vaccine Research Center of the National Institute of Allergy and Infectious Diseases at the NIH. His primary interests are vaccine development for viral diseases, viral pathogenesis, mechanism of immunity, and pandemic preparedness. His laboratory has developed novel vaccines for RSV, flu, Zika, and also coronavirus, including COVID-19. Dr. Graham will share with us his experience on the current state of the COVID-19 vaccine development, focusing mostly on mRNA vaccine candidates. So please, Dr. Graham.
Thank you very much. I hope you can hear me. I'm pleased to be part of this session today, and I really appreciate Mkunde and Florian helping to prepare with such nice overviews. I work at the Vaccine Research Center, which is located on the Bethesda campus at NIH, and our mission is to develop vaccines and antibody products to prevent and treat infectious diseases. And one of the unique parts of this center, and one of the privileges for working there, is that we not only have this basic research building, but we have process development and pilot manufacturing and our own clinical trial system that allows us to develop vaccines and take them all the way to clinic. And I'm going to talk, most of my work is involved either influenza, RSV, or coronaviruses and paramyxoviruses. I'm going to talk today about structure-based design and how it informed what we've done with mRNA vaccines, because we've had a lot of learning between pneumovirus, RSV-F to paramyxovirus-F, and that has informed our work on the coronavirus spike. And just as an example of why it's so important to keep these proteins in their pre-fusion conformation is shown in this video. And you notice how the beginning and the end is so different because of the massive rearrangement that's required for membrane fusion and viral entry. And so the finding we made on RSV several years ago, that this pre-fusion stabilized molecule is much more immunogenic. It's been carried through now paramyxoviruses and and now the coronavirus. We wanted to combine that, what we call precision vaccinology, with a rapid platform manufacturing. And so we've been working with Moderna for the last 3 or 4 years on both paramyxo and coronavirus projects, and had developed an mRNA delivery approach for the stabilized spike of MERS coronavirus. And the way the mRNA works is that a lipid encapsulated mRNA gets into the cell cytoplasm, where it can be translated into protein. And the protein we are making is membrane anchored and stabilized in this pre-fusion conformation. So we've combined precision with speed, and it's one of the reasons we think we got off to a relatively quick start. And we think everything begins with this structure-based approach to design. And so making this protein early on, being able to see that it was in the right conformation, gave us a lot of confidence in what we were doing. And starting with that protein, it allowed us to rapidly develop assays, diagnostic assays. It allowed us to make B-cell probes to discover new antibodies, as we did with Abcelera. And the MAB 555 that Lilly has announced, it came from that collaboration. And it's allowed us to make vaccines. And we're working on mRNA vaccines, and you've heard a number of other vaccine products are also in development. And these are the first 6 that will enter phase 3 trials in the US. And 5 of these 6 are using this stabilized form of the spike protein to keep it in this pre-fusion conformation, both to preserve epitopes at the apex of this molecule, but also because it improves overall expression levels from the cell. These first 6 products include 2 mRNAs, as you heard, 2 adenovectors, and 2 subunit protein approaches that are going forward. So it's a variety of different approaches coming along. And so beginning first with the sequence is published on January 10th, we were able to make protein within 3 weeks, which allowed the structure to be solved, ELISAs, prove immunogenicity, and in phase 1 and trials were started leading to phase 3 trials within 6.5 months. And using these same reagents, looking for antibodies, starting with sorting on February 27th, Lilly was able to take that initial product into phase 3 within 5 months. And as you may have heard just a couple of weeks ago, announced that this antibody, if given to people with moderate, mild or moderate coronavirus disease, will reduce progression to hospitalization. So we've looked at the mRNA from Moderna in both mice, monkeys, and now humans. It's immunogenic in all these species. We've shown that it is protective in the lung and nose or upper and lower airway of both mice and monkeys, and have also done T-cell analysis in combination with the neutralizing activity. We think is evidence that it should be relatively safe, at least free from some of the concerns about enhanced disease. So just briefly to go through some of the non-human primate and clinical data, we immunized rhesus macaques with either 10 or 100 micrograms at week 0 and week 4, and did a challenge at week 8, and looking at the binding activity or binding antibodies, either at the 10 microgram dose in blue or the 100 microgram in red, you see a nearly universal response after a single dose, and all animals responded after the second dose and boosted at least 10-fold. We also looked at domain-specific antibodies, either receptor binding domain or the N-terminal domain, which we think is another important target for protection. And in both cases, the 100 microgram was well above the responses in convalescent sera from a panel of subjects with a range of disease severity. 10 microgram dose was at the upper end of that convalescent panel. Likewise, for functional antibody responses, you can show that the ACE2 binding inhibition activity is well above convalescent sera panel at 100 and at the upper end for 10 microgram dose. And looking at the live neutralization assay using the nanolute or luciferase reporter system from Ralph Barrick's lab, you can show responses, as Florian mentioned, up to 3,500 at the 100 microgram dose level, which is what's going forward in the clinic, and even at the upper end with the 10 microgram dose level. T cell responses are Th1 biased, meaning they're dominantly producing interferon or TNF and make very little, if any, IL-4 or IL-13 responses. And if you do an activation looking for memory T cells, you can also show that at the 100 microgram dose in particular, this type of vaccine induces a strong response of IL-21, which suggests a good T follicular helper cell response. These types of immune responses resulted in good protection of both the lower airway and the upper airway. The BAL, this is subgenomic RNA, meaning that this is evidence of replicating virus. And in the lung, both the 10 and 100 microgram dose levels largely protected or rapidly cleared virus from the lower airway. The upper airway, the 100 microgram dose level by day 2 was either cleared or prevented virus infection in the nose. The 10 microgram dose level was on the border and did clear slightly faster by day 4, but most of the protection that we saw in the upper airway was at the 100 microgram dose. In humans, we've looked at a variety of age levels, 18 to 55, 56 to 70, and greater than 71. The older cohort paper was just published and went online last night, so you can look for that. The injections are given at 0 and 4 weeks. And again, these are either receptor binding domain or full-length trimeric spike on the left and receptor binding domain antibodies on the right. And in the humans, even at the lowest dose of 25 micrograms, all people responded after a single dose, making a spike-specific antibody. And all people boosted at least 10-fold above the primary immunization. The 100 microgram and 250 microgram dose levels were about the same, and because 250 was associated with greater side effects, the 100 microgram dose is what has gone forward into advanced stage testing. And you see also that these responses are either at or above or at the higher end of convalescent panel of sera. For neutralizing activity, either using a pseudovirus nude assay based on a lentivirus reporter system or a live virus classical PRNT using unmodified virus and vero cells, you can show a good neutralizing activity after the second dose. It's either between the third and fourth quartile level, about 3 to 4-fold higher than the geometric mean of convalescent sera. And the same is true for the live virus neutralization assay of being at the upper end of convalescent sera. And just showing you some of the data from that older cohort paper that came out last night, in the 56 to 70 or the greater than 71 group at 100 micrograms, you could show in a fluorescent reporter nude assay, which uses a neon green live virus reporter, that you are inducing responses in the 2 older age cohorts here compared to the younger age cohort at about the same level. So there's not much drop off in the older age groups. So in summary, this product development and clinical evaluation started quickly because of prior fundamental, basic, and translational research that was based on what we call a prototype pathogen approach to pandemic preparedness. We think it was successful because it combined not just the rapid manufacturing, but also more precise antigen design. And it was able to go quickly because of a pre-established public-private partnership. This product is immunogenic and well tolerated in mice, non-human primates, and humans, and protects both upper and lower airways. It's immunogenic and Th1 biased in older age groups. And the phase 3 trial started July 27th and is nearly enrolled. Now, I didn't mention much about the Pfizer product. Florian mentioned it briefly. BioNTech or BioNTech and Pfizer collaborated to make a 1-methyl-uridine modified mRNA product that was either based initially on the RBD only, or then the full-length spike. It was also stabilized with a 2 proline mutation. And so the product going forward into the Pfizer phase 3 and this product from Moderna are very, very similar. I just want to thank our principal investigators at the VRC and program heads shown here, my long-term collaborator, Jason McClellan, who's helped with structural biology, starting with RSV and through NEPA and other projects, including coronavirus, and Dr. Kizzmekia Corbett, who's somewhat hiding here, who helped manage and direct the small coronavirus team we had working on this over the last 4 or 5 years. And Moderna, our collaborators who made the mRNA, Mark Dennison, whose lab Jim Chappell did the live virus nukes, and at UNC, Ralph Barrick and his group did the reporter virus neutralization assays. And I should have mentioned Mehul, who did the fluorescent reporter. So thank you very much, and I'll stop there.
Thank you, Dr. Graham, for this very interesting overview. And in this topic, I think many of us were really curious about this vaccine. We have heard much about it on the press. Not everyone had the time to go through the paper you published, and so this overview was very clear. I also think it was not just the fact of this specific vaccine, but you show us a sort of paradigm of this public-private partnership and a platform that could be theoretically used also for other pathogens. So it's a way of working that may be applied not only in this pandemic. I was also impressed by the data on the elderly, because we know that it's not usual such a high immunogenicity of the elderly. So really very interesting. It's now my pleasure to introduce Dr. Holden Thorp, Editor-in-Chief of Science from 2019. Before he was at the Washington University, and is where he is also currently Rita Levi-Montalcini Distinguished Professor, holding appointments in both chemistry and medicine. Who had the chance to read his editorials will know that he's author of these very interesting and provocative editorials in science. And Dr. Thorp will now share with us his point of view on the challenges in this COVID-19 vaccine rush.
Thank you very much, Francesco. And it's always good to be introduced by someone who has no trouble pronouncing the name of the person who I have the honor of holding a professorship named after, Rita Levi-Montalcini. I just want to tell you a little bit about what goes on at Science when something like this is happening. I'm not going to use any slides, just give you a little overview. Dr. Chachage and Krammer and Graham have done a great job of setting out the science, and it is our honor to support them in the work that they are doing. They are the real warriors here. I have a wise colleague who describes what we do at the journals as being the stagehands of the play. I guess that makes me the stage manager. And it's an honor to do that for science, especially at this important time. Barney and Florian, thank you for all the work that you do, the papers you send us and the reviews you do. It's a huge amount of work, and the scientists who are working so hard right now on this problem deserve our respect, and they're really the people that we try to write for in our commentary section. Most of you would know that science has 3 sections. We have news, research, and insights. And then we have 5 other journals, and all of the journals have published something on COVID, even science robotics. But the others are all either general or life sciences journals. And so we've seen quite a lot of activity as it relates to COVID. We have gotten about 4,000 COVID papers. About 2,500 of those are research. The other 1,500 are commentary. We published about 100 of those papers, which we think is a lot. We're over budget on our pages for this year because of the influx of COVID, but it's been hard. It's harder to get a COVID paper into science than in any other field right now because there's just so much activity. My colleagues who are helping make sure that we create a scientific record that we're proud of are doing an extraordinary job. And as Barney said, we were honored that he sent the spike protein to us. We published it on 9 days after we got it. And it certainly will go down in history as one of the most important science papers ever. So Barney, thank you for thinking of us. We're always here anytime you want to send anything. Thanks for, as you outlined so beautifully, the way that you are positioned in this whole thing. And Florian, I just want to say, if you haven't seen Florian's epic Twitter thread on the vaccines, I encourage you to look at that. It's extraordinary and very enlightening. So on the research side, we've seen a ton of influx ever since we got Barney's spike paper, and we've published important papers on the structural biology of all of those proteins that you saw, and lots of stuff on the immunology and modeling and the vaccines, especially a lot of the monkey trials were in science, and the antibodies. We published the Regeneron papers and a number of the others that have followed along. So that's on the research side. On news, we have reporters who have worked on pandemics, sort of like what Barney was saying, for years, and they have been sort of preparing themselves for this, and John Cohen and Kai Kupfer-Schmidt for sure, but we have a lot of other extraordinary reporters at Science who have covered lots of important things. Just last night, we published a story where John Cohen went with Moncef Slaoui to visit some of the Moderna sites, and I encourage you to read that. It's a really informative story. Then Insights is where we have our commentary. We want to be the best place for scientists to talk to other scientists. And so our perspectives, we've had a number of sort of hypothesis perspectives and many reviews to cover a lot of the COVID area. And policy forums about how to ethically roll out the vaccine and what the federal government in the United States is doing to prepare for all of this. and the number of the traffic that we have gotten to our website on all of these things is a factor of 2 or 3 or more than what we normally get. So we're way over 1 million people a day looking at our website to see all this stuff. Which brings me to, as Francesco noted, the editorials that I've written. I'm an inorganic chemist, so I've had to learn a lot of immunology and vaccinology and other things. But fortunately, I have a position where either I or my colleagues are talking to all the very best people. And so I try to synthesize that and put it in perspective, mostly around the United States and its response to COVID, just because, you know, we are based in Washington, D.C., and we do talk to a lot of the folks who are involved in the United States response, but many of the same issues have played out, unfortunately, around the world in the mismanagement of the pandemic by most of the governments. You know, there are a few that we like to celebrate who've done a good job in New Zealand and a few of the Asian countries, and I think Germany started off pretty well. Let's hope they can hold it. But for the most part, you know, a lot of the same challenges that we've had in the United States have played out around the world. I started speaking out pretty strongly in this kind of critical period, which I think when historians write all this up will be seen as the critical period of this whole story from the middle of February to the end of March or so. You saw in Barney's talk what he and his colleagues were doing then. They were working extremely hard and extremely fast to get us to the point where we could do something about this pandemic, but yet the President of the United States said, for example, do us a favor and speed it up. We knew that that was a ridiculous thing for him to say, because it was implying that we weren't speeding it up, and yet you just saw Barney and Kizzmekia Corbett and their colleagues and many others working on these other vaccines were working fast. So for him to imply that science wasn't going to do everything that it could to conquer this was disrespectful in every respect. You know, when I write my editorials, I think of somebody who is in one of these labs, any of the labs you've heard from today, who works all day trying to do something about this, and then comes home and turns on the TV and sees the president undermining everything they've done. And so I try to speak for those folks, because they are the valiant souls who are leading us to a better place. And so I've written a string of editorials across this time. 10 days ago, when we heard the tape of President Trump talking to Bob Woodward, admitting that he knew all along, even though he was downplaying it, that this was a serious crisis. To me, I thought that was a demoralizing event, again, for these folks that I'm writing for, who have been working so hard to hear their own president in his own voice admitting that he was lying this whole time. I've also been extremely critical of the FDA commissioner. And that's a interesting tightrope, because I think there are a lot of great people working at the FDA, but unfortunately, Commissioner Hahn has blown his credibility multiple times, and I'm very worried about the politicization of what you just saw as a highly rigorous scientific process. The the approval of an emergency use authorization for a vaccine in the United States, or an approval, should be done at the most rigorous possible level. And for the president last night at the debate, which was a shameful and embarrassing moment for the United States in every respect, he, through his own handpicked leader of the of Operation Warp Speed, Moncef Slaoui, who within industry is a highly successful developer of vaccines, threw him under the bus, saying that he didn't agree that what Monsef had said, and I used to work with Monsef, that what Monsef has said and what Commissioner Redfield, our Director Redfield, and others have said that about the timeline of the vaccine isn't correct. And, that worries me greatly. I was encouraged yesterday that 7 former FDA directors from both parties wrote a piece in the Washington Post, echoing a lot of the things that we have been saying about how the Trump administration is undermining the credibility of the FDA. So I will continue to speak out about that. Those issues are important in the United States, but like I say, they're playing out around the world. And it is an honor for me to speak for science and to speak for and try to facilitate the work that you have heard about. And so I thank my colleagues on the panel for the extraordinary effort that they're putting into this. And that's probably enough for me to say right now. And I look forward to any questions. Thanks, Barney.
Thank you very much. I think that as working in the lab, often we forgot that a vaccine has to be then not only discovered and tested, but also produced, commercialized. So there is a big issue of politics, of legislation. And so the point of view you are raising are very important, and every one of us should remember them. So I'm now giving the chair to Mkunde to moderate the question and answer session.
Thanks, Francesco. So thank you again, everyone. Thank you, Dr. Barney, Dr. Thorp, Dr. Florian, for your wonderful talks that we've benefited from a lot, and the current updates on the landscape of vaccine candidates against COVID-19. So let me just remind the audience that if you have a question for any of the talks or any of the speakers, please leave them in our chat box, and we'll try to get to them if we will have time. We do have a bit of time. So let me just start with the questions that have come our way already. There's a question about whether these currently promising vaccines are likely to be protective against the virulent SARS-CoV-2 mutations that might arise, and how the rate of the SARS-CoV-2 mutation is at the moment. Is it something like what we're seeing with the flu vaccine? Maybe if Florian, you can respond to that, if you don't mind.
Yeah, I can try to. So just the response to the first comment. There is one mutation that occurred in the spike that seems there is no definite proof yet, seems to make the virus a little bit more transmissible. What we have also seen, there's now 2 papers, and I think they're still in the preprint stage. We see that those viruses seem to be neutralized a little bit easier, right? And there is a paper in hamsters that shows that, the pathogenicity of the 2 virus variants is very similar. So it doesn't look like that is going to be a problem. And just as a reminder, that new version of the virus already caused a lot of the outbreaks. Even the epidemic in New York was mainly caused by that version already. So I think the situation is very different from what we see with influenza. Influenza is a very special beast. We have a lot of antigenic drift in flu. That's why the vaccines have to be reformulated every year. And that's actually the only RNA virus where we, except for HIV, where we see this. So if you, for example, look at the mumps or measles vaccines, which were developed in the 60s, we were still using the same vaccine strains very successfully. These are viruses that have been isolated many, many years ago, and they still work. And those are also RNA viruses. And we also don't see that much antigenic drift with human coronaviruses. So the situation is very different from flu. We don't expect that we have continuous antigenic drift. And the vaccines, based on the constructs that we have right now, are expected to work also against strains that have some mutations. And, you know, the virus mutates, that it's an RNA virus, there's no question about that. But there's a difference between mutations and escape from immune responses. And I think right now there's not really any big concerns about that. Of course, we cannot predict the future. There might always be an issue in the future, but also the fact that there's multiple neutralizing epitopes on the RBD and then the spike outside of the RBD, and a lot of vaccines include the full-length spike, would help to mitigate any kind of single changes in antigenicity. So it's not a major concern right now, in my opinion.
Thank you. Thank you so much for your response. There's another question that has been posed on what type of side effects are actually being observed right now in the clinical trials that are going on. Dr. Barney, can you please respond to that?
Okay, yes. And first, let me just agree with what Florian just said. The polyclonal response by vaccines is going to be difficult for this type of virus to escape. And there are data now for the 614G virus that he was referring to in the supplemental file of the paper that came out yesterday. If you want to go look at that, it's about 5 to 10 times more sensitive to neutralization than the original 614D version of the virus. The side effects we've seen so far have been mostly the typical things you see in other vaccine studies, with local reactogenicity, some sore arms, and sometimes systemic reactogenicity, especially after the second dose of RNA vaccines. You sometimes get a fever that comes maybe 12 to 14 hours after an injection, after the second injection. But it goes away after about 6 hours, and then people seem to be fine. So it's very similar to what we've seen in other vaccines. For the adenovector vaccines, their reactogenicity is usually dose dependent, and it can often be managed by pre-treatment with Tylenol and things like that. And so at this point, I haven't seen any real important side effects that can be generalized across these vaccines that would prevent any problem from going forward.
Okay, thanks. I have a question to Dr. Thorp. So how do you regulate a compromised regulator in the case of the FDA person that you've talked about now? How will the community be assured that you know, the FDA will actually do its job in critically reviewing the candidates that are actually in the pipeline right now, and to do that without any pressure from the government, or not in any biased form? How do you do that?
Yeah, well, hopefully there are a lot of checks in the process. First of all, like I said, there are career people at the FDA who are extraordinary and important public servants in America, great scientists. They will do everything they can, I'm sure, to make sure that the processes are followed. Then I think there are the companies who cannot afford to have a kind of a misfire on an unsafe vaccine. Of course, they have a financial incentive to be the first ones to get there. And there's certainly a lot of speculation that Pfizer will get data first. Hopefully, they won't apply for an emergency use authorization prematurely. I don't think it's in their business interest to do that. They did sign a letter saying they would follow the science, so hopefully that's the case. And finally, I think there are other extraordinary leaders at the NIH and elsewhere who have staked their careers on this. And like I said, Moncef Slaoui, who you know, was an excellent choice to run Operation Warp Speed. because he did the Shingrix vaccine, which is one of the most successful vaccines that's rolled out in the United States. And all of these folks have pledged to follow the science, and I'm hopeful that they will. I worry that there will be more confusion, and I don't worry, I know there will be more confusion sown by the administration because they sowed confusion about hydroxychloroquine, they sowed confusion about convalescent plasma, and they're already sowing confusion about the timeline of the vaccine and about how it's going to be approved. And, you know, unfortunately, there was a big story in, a big poll that came out yesterday from Pew, which is a big polling group in the United States, showing that there's a lot of trust in science around the world, but in the United States, only 20% of Republicans have a lot of confidence in science. So it's politically useful, and it has been for decades before it was politically useful to create confusion about environmental science. Now it's politically useful to create confusion about this for Republicans because their voters, only 20% of them have a lot of confidence in science. And so that's just something we're fighting in the United States. But like I say, it's around the world. It's in India, it's in Russia, it's lots of places where we have leaders who won't listen to this. So I'm cautiously optimistic that we won't actually get a formal approval, because I just think there are so many people who will rise up to try to stop it. But I'm confident that there will be a lot of confusion about this, and I worry about that in terms of getting enough people vaccinated to stop the epidemic.
Yeah, thank you, Dr. Thorp. Yeah, there is a lot of mistrust, only in the US, but yeah, I think across the globe. So it is kind of sad when you see compromised people fueling into this. So yeah, it's a tough period. It's a confusing time. There has also been a question that has been posed on whether the vaccine, when it becomes available, if it will also, as far as I know, the WHO is committed to make sure that the priority is given to people who need the vaccine, that's healthcare all around the world, the elderly, people with underlying conditions, and for the resource-limited countries, it will be made available for free, while the resource-rich countries will have to purchase it. When it comes to the implementation side, I don't know how it's going to work out, because I hear that some rich countries have already started to kind of bid for the upcoming vaccines or something like that. So I'm not sure how it's going to work out when it comes to the implementation stage. Does anybody want to add on to that? Anyone can take the stage.
Well, we've had a number of policy forums about this. And so there are very smart people thinking, I think, the right thoughts about how to do this. But the political challenge of getting it done that way is daunting, to say the least.
I think the first step is to produce and have available a safe and effective vaccine. So really we have to, and I think the discussion ahead of time to make sure we can distribute it is important, but the first step is to get a safe and effective vaccine. I just want to add to the layers of oversight that are going on, because it starts with the people who develop the vaccine, the investigators, and the companies who do not want to see a vaccine go out that would be unsafe or not work. So some of the oversight begins there. The clinical investigators are independent from the companies, and so there's another level of oversight there. There's an independent data and safety monitoring board that is reviewing the data. So there's another level there. And then the FDA, despite the politicization, as Dr. Thorp said, has a lot of really serious talented, competent people who are looking over this. And even and who've imposed not only efficacy standards, but safety standards that have to be met in a certain length of time for safety follow-up before you can even get a EUA application. And so all of those, and plus the advisory committee for immunization practices beyond that is going to make recommendations for how to use the vaccine. So there are so many layers of oversight and concern to get this right that I think even the crazy politics we're in now are going to be, it's going to be hard for them to make this not go the right way.
Yeah, thank you so much. Unfortunately, I think we're running a bit out of time, so I will only take one more question that is about returning to economic normality before a vaccine is available. How do we do that? Anybody wants to have a go at that?
Well, again, we've published a lot of papers on modeling and masks and all of that kind of thing. I don't know about economic normality, but I think if we could have had, and we still could, a more orderly, logical approach to this, we could be doing a lot more right now than we are. I mean, we do know lots of stuff about environments where the virus doesn't spread, and we know if you do enough testing, you know, the sports leagues have mostly figured it out. I guess the NFL in the United States has a little problem this weekend, but the NBA and Major League Baseball were able to do it. We know mostly environments where people are being careful, the virus isn't spreading. But unfortunately, we don't have an orderly, coordinated, cohesive approach to getting there. And I think the countries that have had that have more economic activity that's going on. And, you know, I'm hopeful that as we get the antibodies and then we start vaccinating people that are vulnerable and healthcare workers, and then, you know, we'll see things continue to improve over the next year. But we're not going to get there as fast as we could if we had an orderly process to do that. Some of the countries in Europe have done a nice job, but from what I can tell, just looking at the data, it looks like some of the increased activity now is bringing the cases back up, and that's inevitable.
Yeah, I wanted to comment on that too. I mean, I think we have some models that we could use, right? And of course, you need to have a government with a plan that is based on science, and you need to have a population that actually sticks to that plan, right? And I think if you look at some Asian countries like South Korea, for example, where there was a huge outbreak initially with 10,000 cases, they got that under control and they didn't have major problems since then. They're having a relatively normal life. And I think, so it's really two components. It's politicians that put the right policies in place and populations that trust those politicians and actually stick to the rules. But as we all know, that doesn't seem to be possible in most countries around the globe.
Yeah, that's an issue. Well, thanks very much once again to all the speakers and all the people who could join us today. Thank you for your time. It was a very, very wonderful session with wonderful talks and a wonderful discussion. Before we end, I would like to welcome my colleague, Sabina, who will give us an overview of the upcoming sessions in case you're interested in joining us in a month's time. Thank you very much.
Thank you, Francisco and Mkunde, for moderating the sessions. On behalf of One Health Knowledge Cafe, I would like to thank all the presenters for the excellent presentation and active discussions. We are really grateful for your time and effort you took took to share your presentation, your knowledge and experiences. Special thanks to all the attendees who attended the webinar. And with this, I also would like to share information about the upcoming webinars. We have a webinar on October 30 on traveling during COVID-19, exactly at 1:30 p.m. GMT. The possible speakers are from Argentina, Nepal, and Mozambique. And similarly, in November, we have planned for a webinar on diagnosis and treatment of COVID-19. On December, health system and COVID-19, and other potential topics include health equity in context of COVID-19. You can have more details on our CIH websites. So with this, I'd like to thank everyone. The resources will be made available in our CIH website. Thank you for joining us. With this, I would like to officially announce the end of this webinar. Thank you, everyone. Have a good day. Stay safe. Thank you.
Bye-bye.
Bye.
Thank you all.
Bye. Thank you.